Come on in. Right, I hope last week's lecture, Cochlear Implants, was as good as I thought that it was. I'll check if it is actually on now, because I think she recorded it, but she didn't upload it, so we'll do that today together with that one, because I did press the record button there, just to make double sure. It's recording, yes. Right, temporal properties. We go through, well, there are three characteristics of hearing. You remember what the perceptual dimensions were? One was loudness, one is frequency, that's right, and the third one, did you remember?
Quality.
No. Localization, that's right. So we'll spend a lecture on localization, we'll spend a lecture on pitch and on loudness, and to bind these together we need to investigate what are the temporal properties. Pitch will have to do a lot of properties with temporal. Well, pitch comes from temporal properties, but there are a few really interesting aspects about the auditory system. The healthy auditory system and the impaired auditory system that we as audiologists really need to understand what's going on with the normal and with the impaired ear. And also a little bit of criticism, what's going on with our investigations and what we can actually do about it. Right, here's the learning outcomes, talking about temporal resolution. And temporal integration as well later on. Forward backwards masking, TMTFs, don't worry about it, this is for reminding me basically at the beginning to organize these lectures. A little bit about modeling temporal resolution, cochlear hearing loss, so all the effects, temporal fine structure, and 2 words about the modulation filter banks. I based this lecture again on the Brian Moore book, but there's also a number of issues here from other books that I will upload one of the chapters from Chris Platt's book, which is really good on the temporal aspects. Right, so we're talking about 2 different things, namely temporal resolution, which is the acuity, versus the temporal integration. So the temporal resolution is our ability to detect a change in a stimulus over time, and the temporal integration is the ability to enhance perception by integrating longer. Both obviously have to do with the perception of time, and both are important aspects about our hearing. We concentrate a little bit we concentrate the first half here on the temporal acuity, and I've got a little demo here from Utsma's CD on auditory perception. This is not a straightforward basic experiment. This is a little bit to explain where the whole issue of perception goes to, which we will explore more in the speech lecture when we talk about auditory scene analysis. And the experiment here, let's do it. It's about a minute long, and it demonstrates gap detection and an interesting perceptual effect.
Apparent continuity. Before playing this demonstration, please read the warning about loudness level in the booklet.
Have you done that?
We present a tone alternating with a silence. Then the silence is replaced by a soft white noise. After each group of cycles, the noise is increased in intensity. In the final group, the spectrum level of the noise is the same as that of the tone. At this setting, the tone seems continuous behind the noise.
Okay, just to repeat what you just said in this nice American accent. Do we? I haven't got anything here. You hear 2 sounds, and there's a gap in between. I don't actually know how long the gap is, 100 milliseconds or something. And then this gap will be filled in with noise. And listen to what you hear, what happens to your perception of the pure tone. Try to ignore the gap, try to ignore the noise. Just listen to what happens to the pure tone. You hear the gap? what you should have heard, is always these experiments, what most people do here, is that the gap disappears. Perceptually, there will be a continuous tone which is masked by the noise bursts, but your brain will make up for the gaps in the tone. That is an illustration of what your brain needs temporal information to create a picture of the outside world. And we'll talk a lot about that when we talk about speech, because speech is exactly that. It's a very complex stimulus that is often not audible for us because it's masked or it's missing, and it's all sorts of things happening to it. And this is one of the very simple illustrations. What happens, what the brain fills in based on these gaps and on the masking. So two keywords here, gap and masking, which we'll investigate now in the next hour or so. So first, the types of masking. The ones that we already discovered is on-frequency and off-frequency masking. So on-frequency means one tone masks at the same frequency as the signal. So like a narrow band noise masks a pure tone. And in practice, this usually means that the masker contains a frequency the same frequency that the signal has. The other one is the off-frequency masking. Maskers at a frequency different from the signal frequency. That's, for example, with the notched noise or the PTC method. But forget these, because these don't really tell us something about the time. We're now talking about the two different ones, namely simultaneous masking and non-simultaneous masking. So simultaneous masking is the one that we usually have, namely its tone at the noise or the tone at the masker are happening at the same time. But they can also be non-simultaneously. The signal can precede the masker, or the signal can follow the masker. So both of these lead to masking. One is called backward masking, one is called forward masking. Here we have the schematic representation. Backward masking, here's the signal, and the masker follows afterwards. Forward masking, first the masker and then the signal. And we also have the masker, signal, masker, which is like a little schematic model of everything that we have in communication. We always have a noise, and in between we have a signal that we want to listen to. You can here read also signal being speech and masker being background noise, but this is the simple way that we model it for our experiments here. Okay, here's an example. Apologies here that some of these slides are not in here. I expanded that a little bit to give you a little bit more demonstration of what we're actually talking about. I hope that works here, because it had some slight issues on my computer. Let's listen to it first. Was too loud? Was good. This is white noise. No, it's not white noise, it's pink noise. And then there's a little gap, 10 milliseconds, and there's a very loud tone. Did you hear that, Pip? Yeah. Easy. This is the same noise, but the tone is here much reduced. Do you think you will hear that? Let's find out. Close your eyes and listen. This is weird. I did this experiment. You probably recognize it hopefully by now in Adobe Audition. Yeah, you should have some handle on that, because it's very easy to generate something in there, like a pink noise and then the gap and then a little tone in there. If you play these sounds on Adobe Audition, you can actually control them. The amplitude and the decibels there are fairly accurate. If I then download them, import them into PowerPoint, PowerPoint will mess around with the amplitudes of my sound. Why? Because it uses compression, which is nice in almost all presentations if it actually makes everything of an audible nice level, but for our purposes it's exactly killing the effect that we want to show. So if I make it a little bit louder, it turns out that PowerPoint will make it exactly as loud as the other one. And if I make it too quiet, which I did here now, on this computer, it will actually completely inaudible. I can't hear it anymore. So I apologize for the problem with PowerPoint here, but you can generate, or if you download it, you probably with headphones you'll hear the sound. But you'll see the effect here, yeah? So at some point this tone becomes inaudible if I reduce the amplitude. And that's exactly what we measure here, which is that we measure the just noticeable, or the just detectable level of a signal presented after a masker. So this is a schematic one. Here's the masker, here's a little gap, and here's the signal. And we have two parameters that we can change to investigate. Namely, one is the gap, the length of the gap, and the other one is the amplitude of the signal. We can also change the duration of the signal. We come to that later. But first of all, the gap and the signal. And here's on the left, what happens if we change the size of the gap. This is what we observe, namely the threshold at which we do hear the sound, and this is the threshold at which the masker is presented. So if we have a very quiet masker, 30 decibel, then we only need a very quiet sound for all of them. They're not that much different. But if we have now, let's say, a proper masker level here, where all sorts of fun stuff like upward spread of masking and stuff happens, let's say 100 decibel here as the extreme, then you see that you need for the 40 millisecond gap, so quite a long gap, you will detect a 40 decibel sound. So that's not that loud. Okay, so it's not the 100, even if the masker is 100 decibel, it will not mask if the gap is sufficiently long. But if we make the gap shorter, so 20 milliseconds or even 2 milliseconds, 2 milliseconds is very short, obviously, we need quite a sufficient level, so 80 decibel or 60 decibel, to hear it. Yeah? So short, take-home message, the shorter the gap, the more masking. Or another word, the longer the gap, the longer you put two stimuli apart, the less interaction between them. Obvious. The other parameter that we can change is the amplitude of the stimulus that we detect, and we change the duration of the gap. It's the same information here, just presented in a different way. And we see that if we have a longer mask, a longer gap, you get exactly what I said. You get less reduction, or you get more reduction of the signal level, so a smaller threshold. Okay. That's for normal people. The interesting question is what happens with hearing impaired people, and where does this actually come from? But two words about the backward masking. Remember, backward masking is the effect that we first have a signal and then we have a masker. So we make shh. You wouldn't expect that the masker afterwards can actually mask. And energetically it can't. Energetically, it already happened on the basilar membrane before the masker comes. So auditory nerve fibers will have picked up on that signal. But there is an effect of backward masking, which is not well understood, because it doesn't happen on the basilar membrane. It happens more centrally. And everything that happens centrally can be influenced by practice. And it turns out that practice subjects often show little or no backward masking, so you can reduce your own backward masking by training. And therefore I'm not going to investigate backward masking very much, because this is more a psychological effect rather than a physiological effect. But it does exist. Okay. How do we measure temporal accuracy? Now we had one, namely what is the gap, the duration of the gap that I need in a stimulus. There are two ways of measuring it, actually three ways. One is gap detection, and I'll talk about that. What we just discovered was forward masking. So how loud does the stimulus have to be after a short time so that I can still hear it? The second one is I want to measure how small a gap can be so that I'm just able to detect the gap. Okay, because gap detection becomes important when we come to speech perception, because gap detection is, well, not the detection itself of a gap, but the detection of a stimulus in a gap will become the most important feature of speech perception. Right, so gap detection refers to ability to detect a short silent gap in an otherwise continuous signal. It is different to forward masking, but related, because it's the same effect. Because if you have forward masking, obviously, then the gap itself will be masked. Although it's not a stimulus, it's a gap, but it can be masked as well. So, gap threshold is the shortest gap that can be detected, and it depends on the frequency and the type of the stimulus, and on the onset-offset characteristics. It detects on a lot of things, and it's quite difficult experimentally to measure. But it's usually used as a measure of temporal resolution ability, because the experiments itself are very straightforward. Coming to ringing in a second. I'll just give you an example here from a student experiment from a website, which explains pretty well and makes a good circle back to a few lectures ago. The method here, sound 1, sound 2, sound 3. So shh, 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 shh. There was a gap. Now you can ask a number of questions. Let's design an experiment how we measure the duration of the gap that we can just detect. So we could just play either sound 2 or sound 3, and we asked, is there a gap, yes or no? So you say, shh, shh, was there a gap? You say yes or no. If we do that, we get, here's a perceived gap, a correct perceived gap, and this is the duration of people, I think over 50 undergrad students or something, which is fairly normal for an experiment like that. You see, if you're above 4 or 5 milliseconds, you get always correct response, and you get a threshold of about, so let's say 75% criterion, and we draw a line through here, it's about 3 milliseconds. Now let's do the same stimuli, but we do a different experiment. Namely, we ask the question, we have 2 alternative force choice, so we've got stimulus 2 and stimulus 3, and we ask which of the 2 have the gap. So we force the subject to make a choice between the 2, and then we split these 2 up of having the gap in either the first one or the second one. Okay, I could average these and get a realistic answer, but I can also just plot them as having the gap in noise 1 or having the gap in noise 2. And you see that the results are different. And both of these in itself are also different from the first one. So same stimuli, same question, same perception, same listeners, completely different answer of what the response of what the gap detection threshold is. Now, here we go. That's what signal detection theory is about, and that's why it's so important to understand why we are measuring, or what we're measuring here, and why it's so important to know what we define as a threshold, and it's important to write a method section that lets people understand what exactly you did as an experiment. Okay? So just number-wise, if you put 75% in here, it's about the same. Here, it would be between 1.5 and 2 milliseconds rather than 3 milliseconds. So luckily the ballpark figure of 2-3 milliseconds is correct, and if you look in the literature you always see gap detection, we're able to detect about 1.5 millisecond gap roughly. Okay, that's about the take-home message, which is really, really short. Yeah, that's the main point. But that's not, we can do much better than that, and we'll come to that with the modulation transfer function. But first let's explore why we cannot detect any kind of gap, a millisecond or half a millisecond. What happens on the basilar membrane? For that we need to understand a little bit about basilar membrane, which we do know, we're expert in that, and about filters, auditory filters. And I've plotted just 3 auditory filters here that are centered at the same frequency and have different bandwidth. So this is just schematic. That one here, for example, the red one has a bandwidth of say 150 Hertz, the green one 300, and the green one 300, the blue one 600 Hertz. So these are different filters, different bandwidth filters. And if you put a impulse into these, an impulse is that. So your basilar membrane now produced an impulse response to that impulse. That's a standard way of characterizing a filter. Especially linear filters are 100% characterized by them. We know that the auditory system is not linear, the filters are not linear, but we pretend for all intents and purposes that's the best starting point to investigate them. We say, let's assume they're linear. What happens? We get a really good picture of why we cannot detect gaps. Namely, what happens if we put a very short tone into one of these narrow filters is that the basilar membrane is switched on, and then it rings over time. That's this ringing. Every system in nature rings. We call that usually, you call that the impulse response or resonances. Everything has resonances. Even, yeah, you hear the difference? Yes. Why? Because they're different resonances. They're different materials, different size, different everything. And so the basilar membrane as well. It's very, depending on where you are on the basilar membrane, it depends. It produces different ringing, and this ringing cancels out gaps. So if we have, for example, these are narrowband noises in a low frequency, low frequency noise in the same frequency channel, but we have a gap in here of 10 milliseconds. Here as well, 10 millisecond gap. But if we now simulate a filter, an auditory filter, say, and we send this signal, this is an acoustic signal, and this is the signal that we would hear on the basilar membrane because it has gone through the auditory filter. And you see what happens to the gap. It's filled. It's not visible anymore. You have a reduced amplitude in there because nothing comes in, but the filters keep ringing for a while. Keep ringing for a long time, actually. Here, 10 milliseconds is easily a time that can be that they carry on ringing. Obviously, the lower the frequency, the longer the ringing, because the low frequencies have longer wavelength and they will ring for longer. Yeah. The third way of detecting or to describe temporal accuracy in the auditory system is by the way of amplitude modulated sounds, which are signals, for example white noise or pure tones, that are modulated with the specific frequency. So here's the picture, as is a schematic picture. We've got the pressure over time, that would be a pure tone, and this is a modulated pure tone. Now, what we can vary here is two things, namely the modulation frequency and the modulation rate. And what we usually measure is we measure the modulation depth at a specific frequency to find the smallest index of modulation that is detectable. M equals 1 indicates 100% modulation, M equals 0 indicates no modulation. So this would be M equals 0 because it has no modulation, and this here is about 50% modulated because the size between, or the difference between the peak and the valley here is about half of their height. Yeah, just guessing here. But there are two ways of describing that. One is in percent, the other one is in decibel. Both are equivalent, are connected to each other via the logarithm, but on the next slide what you see is the logarithmic values, but don't get afraid of that, this is the same thing. So we plot the M, this modulation index that is just detectable, as a function of the modulation frequency. That gives us the TMTF, temporal modulation transfer function. And that shows that we're very, very accurate in these things, namely much more accurate than a gap detection. So this is not necessarily very different from a gap detection. You just get a gap here, and a gap there, and a gap there, and a gap there, of course, yeah? So you get many gaps. So what this is the whole concept of temporal processing in the auditory system. You don't get one gap, you get 10 of them, and they all look the same. So what is your auditory system doing? It's utilizing this, and it has these multiple looks, because it can measure now 10 gaps and somehow integrate or somehow compare them and gets a much better perception of it. This is why almost all of our stimuli that we produce for communication are actually periodic, which we will discover next week, to give our auditory system many, many more presentations of it. Okay, and hold on to your seats. Compared to vision, we are 3 magnitudes better. What we find out is that we can detect modulation frequencies up to about 1000 Hertz. So 1000 times per second. What's the highest modulation frequency that we can detect in vision? It's about 20 Hertz. Because if you modulate something, or if you change a picture 20 times per second, you don't see the individual pictures anymore, you see a film, as we all know. If you hear 20 times a sound per second, you would still hear every individual sound. You can hear that up to 1000 Hertz. So this is really, what is it, well, 2.5 orders of magnitude. But let's look at the data, and here's some examples as well that I've put in there. So, what we see here is data, Bacon VMeister 85, 1 Hertz, 10 Hertz, 100 Hertz, 1000 Hertz. Well, I said after 1000 Hertz it disappears. We don't hear the difference, the modulation anymore. But here's one that I'll just play as an example. Remember what I said about PowerPoint messes up your stimuli? You should have heard that much more. PowerPoint is actually compressing our amplitude, apparently. I'll be wondering if I can find out what it actually does, but it is not what these wave files sound in nature. Anyway, just here you hear the modulation, I hope, still. Yeah, 1000 Hertz tone modulated 10 times per second. And I made it that is 100% modulated, but this is not 100% modulated. This is more like, I don't know, 50% modulated. Anyway, this is at 0. So 20 times log 1 being 100% modulated would be 0. So 0 dB is the same as 100% modulation. This is the way that people choose to plot this. But don't get carried away from that. It's the same as in percentage. So 50% modulation would be, I don't know, somewhere 12 dB or something like that. So the higher it is up here, the less modulation. So everybody here would say, yes, we can hear the modulation, I hope. But up here, you probably don't hear it anymore. So in a controlled environment, people would still see it, would still hear it. Only up here, they wouldn't hear it anymore. So, first of all, what we observe is this would 1000 Hertz tone 10 times modulated per second. Let's see what happens if we modulate it 100 times per second. You definitely hear that, yeah? It actually becomes something else, this tone. It gets a pitch, which we'll talk about next week. But you definitely hear that this sound is very different from a pure tone of 1000 Hertz, yeah? But if we go higher up here, you probably have problems to differentiate that from a pure tone. That is just above the threshold, so we cannot hear it. And the pure tone itself, just as a comparison, would sound like that. Okay, so a few examples. You see increasing frequency, increasing modulation frequency. That's not the frequency of the tone. Yeah? The tone frequency is called the carrier. We're talking about the modulation frequency. We're very susceptible to that at low frequencies, everything up to say 100 Hertz, and then even 200, 300, it sort of breaks down at around 1000 Hertz. So that covers the whole range of fundamental frequencies in the human voice, and it covers specifically also all the speech components of prosody, of all the information that the low frequency information where speech is located. Okay, so take home message: we're very good at detecting temporal properties in sounds, and we can measure that with at least 3 ways. First one was forward masking, so we can measure thresholds for forward masking. We can measure gap detection, or ultimately we can measure how much a tone is modulated for us to perceive that it is modulated. Well, what good does it do? It allows us to understand glimpsing. Glimpsing is a phenomenon, or is a concept of how to understand how we understand speech and noise. Let me talk you through some of these figures, what is going on here. This is the standard sentence. I think it's a BKB sentence, something like the clown had a funny face. If we put that into noise, in the waveform, it looks something like that. Okay, because the noise now masks at a low level, so this would be a positive SNR, so the noise isn't actually covering all of the speech. You still see the peaks, but you don't see the valleys anymore. You see, this is what I mean with modulation. This is a low frequency, very low frequency modulation, because it goes up and down up and down over the time course of 1 or 2 seconds. So we could calculate that as 1, 2, 3, 4, 5, 6 times in 2 seconds, so 3 times in 1 second. So it's about a third of a hertz. That's roughly just eyeballing what the frequency of the envelope of speech is. That's what we communicate with, yeah, the lowest modulation frequencies. And exactly these, where there is no energy in the speech here, this is all covered by the noise. So we would end up only hearing everything which is above the noise level. Now, this is the spectrogram of these. So this is a spectrogram of a pure speech, this is a spectrogram of a noisy speech, and you can see immediately the problem. Even, well, not as bad as here, but you see that the noise covers all of the glimpses, or all of the gaps in here. This is what we call a glimpse, or a gap, because there is a gap between this one and that one. When I talk, there are lots of gaps that I make between the words or between syllables for you to be able to understand that. If we have the noise, it fills up with that. Now, there is a very efficient way, the most efficient way of signal processing that has been developed so far in the last 5 years or so, which is called a mask, a binary mask, in order to recreate or to get rid of the noise, because that's what we want to do in a hearing aid in the end. So this is the research topic of 2014, how to apply this binary mask onto the speech. What is a binary mask? Well, we measure for each of these areas here if there is more noise or is there more speech. And then we decide if it is more noise, we give it a 1. If it is more speech, we give it a 0, or the other way around. Okay, and that gives us the mask, which is called binary because it can only has 1s or 0s. And everything in here that is white, for example, would be an area where we have more speech. And everything that is black would be an area where we have more noise. And if we apply this mask, or looking through a mask mean we'll only look at the bits that are white in here. And if we apply this mask, for example, to our spectrogram in here, we can recreate the speech without much noise. So this is the by far most efficient way of reducing the noise in signal processing schemes, but it has one problem. If you're clever, you might have noticed that. What is the problem? You need to know for the ideal situation in advance what the noise is and what the speech is. So it's a bit circular at the moment. It has been shown as a proof of concept that it can be used, but in order to actually create the mask, you need to know the noise. And in the real world, you don't know the noise because you don't know what is the speech and the noise. You only have both together. So it's a bit of a circular argument at the moment, but we're all hoping that we can do something more of that, because the good noise reduction algorithms are all trying to estimate the noise. And if you know what the noise is, you can reduce the noise using this scheme by enhancing the speech. But we'll come back to that in the speech lecture or in the hearing aid lecture later on as well. But this is just as a motivation why these temporal aspects are also important, because all of these gaps in here allow us to hear something. And here's the extreme example, or the not speech related example, namely this is how glimpsing is defined, or how glimpsing can be made objectively measurable. You see a difference between these two pictures. Where's the difference? You see here it's a little bit wobbly. So what's the stimuli here? The stimuli, this is an amplitude modulated masker. What is it? Signal frequency 1800 Hertz, and we have a signal frequency 1800 Hertz and the masker frequency 1000 Hertz. Now here we have them on top of each other. The problem is the masker level is much higher than the signal level. So let me just estimate that. It's about 10 times higher, which is about 3 dB. So this would be a signal to noise ratio of minus 3 dB, because the masker is 3 dB louder than the signal, or 10 times more energy. So challenge you now, visually, do you see the tone? If you just look at the first half here, Obviously not. This is completely impossible because the masker is 10 times higher than the signal. It completely washes out the signal. But if you now modulate the masker, you produce glimpses. You produce areas where the masker itself has very little energy, and here you give the listener a chance to actually hear the signal. Yeah? So this is what's called listening in the dips, or listening and glimpsing. And this is the sole reason why we're able to hear sounds at very negative or very, very bad SNRs. So we can understand speech to reasonable level at -10, -15 decibels, where the masker is 100 times louder than the signal. And for that we need to understand, or we need to investigate what these temporal aspects are. Obviously there's all sorts of masking going on, because this masks here energetically on the target, but there's also forward masking involved in all of these things. Okay, and the culmination of the whole thing is, and I'll only speak briefly about that, co-modulation masking release, because everything that we talked about so far was happening in the same frequency channel, more or less. 1000 Hertz, 1800 Hertz is still in the same, roughly same frequency level. Well, we pretend it's in the same frequency. So it's on frequency, simultaneous or forward masking. But we have to investigate also what happens if the masker is away in frequency from the signal. So off-frequency maskers over time. Now we bring it all together, and we get to a phenomenon which is called co-modulation masking release, which is a very, very long and complicated word for an actual relatively simple thing, but it's because it contains all of these issues like time and frequency and different frequency bands. It's quite difficult to draw a schematic picture of that. I think this one is probably the best one. Let me talk you through it. I should have made a sound example of that. So here we have, for example, a 700 pure tone, which is buried in a modulated 700 pure tone, so a 700 Hertz narrowband noise. I think we had narrowband noise. You should have a picture in your head how a narrowband noise sounds. It is a very, very good masker. We found that out a few lessons ago when we talked about the filter bandwidth, the filter shapes. But you know, at the best frequency, masker are most efficient, obviously. So in other words, it is virtually impossible to hear the 700 Hertz tone if it is masked with the 700 Hertz narrowband sound, narrowband noise. That's why this face is unhappy here, because it doesn't hear the target. Okay, now let's take the same sound, but now we play other sounds as well. We play even more maskers, but these maskers are in different frequency bands. So we've got a frequency band here at 400, at 500, 900 and 1000 Hertz. And what is the same in all of these? They all have different center frequencies, so they all have the sounds that they played are all different. So it's 400 different tones, but what's the same between that one and that one and that one? The modulation, the envelope. Yeah, the envelope. Here we have the first time the word envelope, but envelope is the same as modulation. It's modulated. So what happens to the perception of this sound? What do you believe? Well, looking at the face, he can hear it. Now this is fascinating. This is really fascinating. Here, a sound that is in itself inaudible, we add more masking, the sound becomes audible again. How can that work? Well, there's a lot of research been done on that, and it is the next step, let's say, from our simple psychophysical experiment where we investigate frequency and time, because here we put it all together in an actual perceptual relevant way, because all stimulus in the real world are wideband. And all stimulus that I produce as speech are not only wideband, but they're also modulated in all frequency bands. This is what speech is all about, and we'll investigate a little bit more about next week when we talk about pitch, because these talk about modulation of pitch is a similar thing, but they make, if you modulate everything outside of your target area, you will reduce the actual masking. That's why it's called masking release. Let's talk about the word. So you have a masking, you release the masking, and how do you do it? By co-modulation, by modulation in different frequency bands. So here's the other way of showing it. You've got the little target in here, which is this black arrow, and you've got the red one is the masker, which is much louder than the target, so you wouldn't hear it. But if you put all of these, it's called flanking bands also, because they flank it here and they flank it there, and you put all of them together, you then get a release. Here's some original data, CMR, co-modulation masking release, so it's the same word. Just look at the top left one, and I'll talk you through what it means. So this is Schoenfeld and Moore. Verhey has done a lot of work on that recently as well. So we have our on the x-axis is the flanking back frequency divided by the signal frequency. So at 1 they are having the same frequency. So that's when we have the most masking. And on the y-axis we have the signal threshold. That's the threshold for detection. So that's the higher that is, the worse is the masking, the most amplitude, the most energy you need in the signal to actually hear it. And you see, let's take the circles here. At the center frequency, that's the case when you have the highest masking. And if you now put additional bands left and right, and you not correlate them, so you don't have actually any modulation at the same time, you get a little bit of a masking release. So the masking release is the difference between that value and that value. But if you correlate them, that's what I mean with modulated at the same time, then you get a massive masking release, especially if they are from here to here, something like 20 decibel or something masking release. Now the fascinating thing is you can also do that dichotic, so you don't actually need to have them in the same ear. You can play the masker on the one ear, or the flanking bands on the one ear, and the masking band on frequency on the other ear, and you can still somehow centrally integrate these two and make sense of that. How that works is up for anybody's guess, but it's a nice observation that triggers new research. How does that work? Okay, 5 minutes about modeling that, because the model actually explains a few things and makes a nice picture of what the effects of hearing impairment actually are. So, that's a very simple model. You recognize that from Brian Moore. I just copied it from his book. Block diagram of the model of temporal resolution. We start with the stimulus, so for example our 700 Hz tone and a 700 narrowband masker. We model bandpass filters. Now guess what? These are the auditory filters, obviously, that we talked about so often. We measure, or we model a very simple nonlinear device. Can you guess what nonlinear device that is? That's the compression in the basilar membrane, our typical compression curve, because that turns out to be the most important part of this model. We have a little temporal integrator that I'll show in a second, and then we have a decision device, which is in the real world our brain, obviously. In the modeling world, it is just something that tells us that is perceptible or not perceptible. That's a very nice way of modeling something. You just make a very simple yes/no answer. Is it above threshold? Is it below threshold? Let's look at some of these examples. So I don't talk about the bandpass filters and the nonlinear device because that's all 2 or 3 lectures ago. That's exactly what we talked about. This is the temporal integrator now. And it's worth looking at that a little bit. So this is the shape of this, what's called a sliding window. What that means is, now you send the sound. Let's say we say simple. Let's just take this one as an example. There's a click, so an impulse. Yeah, my impulse. This is the impulse response. How does that happen? Now we take the impulse, we send it through our auditory filter, then we send it through our compressor, which changes the amplitude. And then we send it through our sliding window. So we apply this window at every point in time, And at this point it obviously has the highest value, and then it falls forward and it falls backwards. Obviously measuring forward and backward masking. Forward masking, because this is a sliding window, it is turned around in time, but don't worry about that too much, because it's basically what it does mathematically is a correlation between the input and the output, the input and the window function. This is the forward masking, and this is the backward masking. There's a little bit of backward masking, but not very much. Forward masking is more pronounced. So, what happens to our click, our impulse? It becomes something like that. That doesn't mean anything at the moment, because everybody perceives the click the same, roughly, hopefully. But what is interesting, if we do a little bit more exciting experiment, well, this would be the response to a step. It's called a step response, and it grows, and then it grows a little bit, and then it falls down. Okay? Now here's an interesting one. This is our gap detection experiment, because we can model that now, auditory filters, nonlinearity, sliding temporal window, and you see that this gap becomes much smaller, because the averaging that happens. Well, because of a number of things. Because of the compression and because of the averaging in time. And if we do that more and more often now, here we have a modulation now. We've got not one gap, we've got two gaps, and you've got many, many gaps. You see that this becomes more and more compressed. Well, there is less and less modulation. Yeah? So our decision device, and we've got a decision device. The decision device is us looking at it and saying, the more modulation we have, even if they all have the same modulation depth, the less actually comes through in the envelope, and the less chance we have of detecting that modulation. Okay? That gives us a nice model, or a nice way of looking at the modulation and deciding, if we have a stimulus now, would we hear it or would we not hear it. Okay, now 3 words about hearing impairment, and then we make a little break. So, all that we've said now indicates that we need to understand about our auditory filters, because there is a thing between the filter bandwidth and the ringing. Remember that? What was it? The sharper the filters, the longer the ringing. The wider the filters, the shorter the ringing. Now here's the conundrum. If you're hearing impaired, or if you have a hearing impaired person, the filter shape become wider. We always said that's the bad thing, because you've got less frequency resolution. But for the temporal resolution, it would mean that they have a much better temporal resolution, because they don't have as much ringing. Is that true? Well, with that question, I'll leave you into the break, and let's meet again at 12, okay? And then we answer that question.
Here. Yeah, it's good. How was the
And then Toronto, Montreal, there's
oh, right. There's not.
The thing about Montreal is that you have to walk up.
I mean, did you use that like after Apple Python?
My friend had 30 steps to get into
I don't know, like, how long, like, I was really tired at this point.
I think she had 15 outside and then you'd open the door and it's just what they're known for. I guess they're very sick people.
Oh, the hearing.
Okay, good idea.
And when I'm looking at
It's too much.
It's too much. I feel like when I'm writing it, there's not much discussion in it. Very much.
Yeah,
It's just like
description. No, be careful about not being too descriptive. Maybe it's still time to concentrate then. I've said to others that it is not a good idea to try to, because it's too wide, the subject. Because you know right now, so because you worked 2 weeks on it, so maybe you can, it's still maybe possible to concentrate. So one of the factors that cochlear uses content, and because you know that talking about everything is probably becomes too descriptive.
yeah, that's what I felt like. So maybe I could just focus on a topic or two because there's not much.
Yeah.
There is mostly animals.
Okay.
Yeah.
You can I've rarely seen a good essay that is only focusing on cochlear fusion. I think it is very interesting and it's possible and very critical, but because it has so many facets, you must be very careful of not overblowing the details, but to actually find a few papers, as you said, there are very few papers, and there must be an essay, that actually investigate what cochlear fusion is about, so not noise and not the ototoxicity, but what's on top of that makes the cochlear fusion. So a simple question, if you never have any noise exposure, never have any ototoxicity, you still suffer from cochlear fusion?
Yeah.
I don't know the answer to that, but I have a guess, but if you discuss that and explicitly take out the ototoxicity, well, you can write it and say, I understand that, and say, okay, there's not the space here to discuss that, and there's not the space to discuss hearing loss. What is left? And that is very interesting. There is a PhD in that question. What is actually, and we've got some things here as well in the next lecture, is what hearing loss is and what it isn't.
Okay, so that was something that I could. That wouldn't be exactly the Islamic country.
Yeah.
You have to describe every single.
No.
Within reason. But I want to say to concentrate rather on, if you find one, two good papers on cochlear fusion, if you describe critically what these two papers say, I'm very happy for you to not discuss many other issues.
I could be wrong, but that would be.
I know it's difficult. I know it's quite. But
if not, I'm just
The university is.
I'm sure you've worked 2 weeks on it. You learned probably quite a lot about it. So the learning outcome is key. Now you just need to be able to summarize it and go back to your learning outcome and summarize all of that. Because there's always a good nothing is easy. Really? There wasn't the research to look up for that. Okay, I appreciate that.
Because I was trying to come on, but I didn't follow all. It was mostly about research, how to work. I saw your videos on
Can you remind me? Can you send me an email? I'll send you a last year's project report so that you get a good idea. I'll send you a really good one, and then you can see what is expected. I don't want to do that to everyone because then they will become very, very serious. But I do understand that the predicament that we have in the
I mean, he would be adamant, no, you should do this. Something would just require. Oh, no. What are you you see the point? Because for example, like some people might start a conversation and go, oh, I saw that or something. I really like that. So that it can be quite helpful.
Oh, okay. Here, okay, that's why.
Like it's nice tea, just nothing to well until about 2 slides and then I start to lose myself. Probably a good time for my brain.
No, it's helpful. And it's like the very excited. Oh, really? So you should go there and
Shall we carry on? The lights go off, the curtain opens. That's your cue to become silent. Right, so I left you with the question: hearing impaired people should be better in temporal resolution than normal people because they have wider filters. And to some evidence that is indeed true, and I'll show you some evidence in a minute that it is true. But in many other situations it is not better, and we need to understand that to understand why hearing impaired people suffer speech and noise. Okay, here's first of all some evidence that shows the effect of, well, presbyacusis on the gap threshold. And there are 3 different groups. There's a very important paper here. Didn't say it, 1992 already, Mark Lutman. 3 different age groups in 1, 2, 3, 4, 5, 6 different hearing threshold level groups. So at 2 kilohertz, we've got young, medium, and older people with no hearing loss, severe hearing loss. And what do you conclude if you're looking at these 3 lines? there is not so much an effect of H. There is a little bit going up and down, but on average, the three H groups look fairly similar. However, there is a drastic effect, well, there is a definite effect between the hearing threshold level and the gap threshold. Sorry, I should have said what was measured here is the threshold. I can't remember exactly what was measured, but it is the threshold of a gap. Remember, the actual number is not so relevant because it really depends on the way that you measure it. But the relative effect, of course, is important. And you see that the effect of the hearing threshold loss is more pronounced than the effect of H. And here's the concurrent result. It's just plotted all of the subjects together, and they measured the gap detection threshold in milliseconds, whether the frequency resolution. Not quite sure what the measure was here, but it is a measure that measures the lower it is, the better your resolution and frequency is, and the shorter that is, the better your gap detection. And all of these people together, it's elderly, so what was it, 50 upwards, are clustering around a straight line here. So there is a linear correlation between the resolution, frequency, and the gap detection, but it doesn't really affect your speech recognition ability. Certainly not in quiet. Only if you have really bad, poor gap, so 40 milliseconds and above here, it will affect your speech recognition. So the take-home message is the frequency resolution declines with the hearing threshold level, but not necessarily with H. So the question of how important is H is a really, really good one. And I don't have an answer for that, but the data show that there is a good correlation between all of the frequency resolution and gap detection and the hearing threshold level, especially at 2 and 4 kilohertz. If you know somebody's hearing threshold at 2 kilohertz, you have a quite good idea what their gap detection, so their temporal properties and their frequency resolution properties are. So you know more about that than when you say somebody is 65 years or 75 years old. The age is not a good predictor for all of these measures, but the hearing threshold level is. Yeah? So just because you become older doesn't mean that you have worse gap detection thresholds. Okay. However, here's one of the results. That's Brian Moore, 1998 paper, that shows now how much worse are hearing impaired people actually compared to normal people when it comes to gap detection or forward masking in that case. So the stimuli here was a 200 millisecond noise, so it goes from here to here. And what was measured was the detectability of a short tone, either at the beginning, in the middle, at the end, or then after a short gap after the noise. So in here it is masking at the same time. So this concurrent masking, and this here is forward masking. And we've got three curves, impaired, normal, and another normal, at 1000 Hertz. So and these were actually people that there's a nice kind of people that have a hearing impairment on one ear and no hearing impairment on the other ear, so you can really nicely match them in amplitude. And I don't know how many people were averaged for that. I think only a small number, 4 or 5. So what you see is that the normal ear shows this normal forward masking curve. So you've got the prediction of when you hear the threshold during the tone, and then after the tone you've got this decaying. So the tone, the bigger the gap becomes, the easier it is to detect the tone. If you do the same experiment at the same sensation level, you would get something like that. So the hearing impaired person would have much sorry, I should explain it the other way around. If you do it at the same SPL, so the same level of tone, you would get the results in the square. So you get the same thresholds for the perception during the noise, but then you get a much sharper effect of the forward masking. Okay, so the hearing impaired person will suffer more from this forward masking than the normal person, but only if you look at the same, if you play the sound at the same SPL. If you play it at the same sensation level, which of course is much louder for the hearing impaired person. Let's say you've got a 40 decibel hearing loss, you will make it 40 dB louder, they have the same sensation level, and then you get this curve, SL, or explain it wrong again. If you do what I'm saying is, if you do the experiment with the people comparable, so either you reduce the loudness for the normal person or you increase the loudness for the hearing impaired people, then the results will be much more similar. What's happening here is that the normal people have been reduced, so they only play it at 60 decibel rather than 90 decibel, and the forward masking curve here is very similar to the forward masking curve of the hearing impaired person. So, take home message. If you compare normal people and hearing impaired people at the same stimuli, hearing impaired people are much worse. If you compensate for the level of loudness, they're actually as good. Or almost as good. So, take-home message is, it's actually not as simple. Hearing impaired people do not necessarily have a reduced temporal resolution, because their filters might as well be much wider. They've got maybe even a better. And let me show some results where they do show better frequency, temporal resolution, namely in the temporal modulation transfer functions. We've shown that figure. Remember that this is the modulation now as a function of the modulation depth, and the higher the better. And we've got the impaired, we've got normal, and we've got normal at SL. And you see that the impaired, these are three different listeners and the average. Just look at the average here. You see that the triangles are almost everywhere above the normal or the normal at sensation level, meaning the hearing impaired people detect modulation at a smaller modulation depth than the normals. They have a better modulation detection. And why that actually is, I can maybe explain very quickly is all due to compression. I should have made a, you remember this curve? Compression curve, input versus output. That's the normal. The hearing impaired person has a linear compression curve. Yeah? So linear means this is the hearing impaired person. If you put 40 decibel in, you get 40 decibel out. Let's say this is 80, let's go two numbers in here, 80, 80. Okay. Whereas the normal person compresses all of that into a much smaller range, a wider dynamic range, but less steep function. So if you have a modulation now in there, let's say we have our, this is the term, modulated. And what we're measuring is if somebody can perceive this dip, this gap. Okay, if you present that to a normal person, you will start, you have the top of the gap is here, let's say, and the bottom of the gap is here. Because you've got a lot of input difference, but the output difference is tiny. Whereas the hearing impaired person would have this as the input, the dynamic range, and has this much difference. Yeah, so this difference versus this difference. So because of the compression on the normal ear, our perception of the modulation is worse than if we had no such compression, a linear system. And since hearing impaired people have a much more linear input-output function, they actually have a better detection of modulation. They can, as shown in these figures. There's only a few people, and the effect isn't large. Let's talk about 5 dB or something, but it's still there. So here, shattering the idea once and forever that hearing impaired people are worse at everything. They're not. They're better at one thing at least. Unfortunately, it doesn't help very much, because in order to have that effect, you need to be above threshold, and you have, because of recruitment, you haven't got much dynamic range. So a clever hearing aid should be able to push everything up into the audible range, but then it compresses that again. So the hearing aid itself compresses that. That's the same effect that what my bloody PowerPoint does here with my stimuli. It actually decreases the compression. Well, the compression decreases the difference. Anyway, but that effect exists. Right, which brings me to the next topic, and I already have that here. I always talked about that, modulation and the fine structure. Let's investigate that a little bit further. There will be more about that in the next coming lectures as well, because this temporal fine structure, or the relationship between the temporal fine structure and the modulation, gives us a few good hints of what happens with hearing impaired people, and maybe what we can do to help them as well. And here's just an explanation, basically just a definition what the temporal fine structure and the modulation is. So the temporal fine structure, here's the three different channels, 300, 1400, 4800 Hertz. That's effectively the movement of the basilar membrane. That's what we call temporal fine structure, because this is as fine as it gets in each frequency band, the movement of the basilar membrane. And if we now, there is a technical way of defining that, but the non-so-technical way is you squint your eye and put a line over the top, you get the envelope of that. Yeah? The technical way of defining that is called Hilbert envelope, but don't worry about that. You can easily just draw a line over the maximum and you get an envelope. So you get these two effects, fine structure and envelope. Both play an important role for our perception. How? Let's investigate a little bit about physiology. I haven't talked about physiology very much in recent lectures. But how does it actually work? How do neurons code this fine structure and the temporal and the envelope? Because that's at the basis, of course, of all of what we're saying. So, how is a frequency coded in the auditory nerve? We have two ways of coding that. And the first one is, going back, is just looking at the right frequency channel or the right auditory filter. So if we have a neuron in the 300 Hertz channel, it will know that it responds to 300 Hertz. That's called place coding. And we'll explain that a little bit later next week, but it's very straightforward. It just means if you know where a neuron is on the basilar membrane, you know which frequency it's tuned to. Remember that each nerve fiber only has about 10 nerves to each hair cell, and that's actually not as very important at the moment. Within the auditory nerve bundle, outermost fiber carrier has frequency. Don't worry. What I'm going to talk about is the temporal coding, not the place coding. So let's concentrate on the temporal coding. Got a little video here that shows what we probably all suspected so far, but we haven't really talked about, is that when neurons fire, they will fire only at a specific point during the stimulus, namely where? When the stimulus is at its maximum, has a maximum amplitude. So again, listen to that, it will fire now. Not down here at the valley, but here at the peak. Yeah, unless it's spontaneous activity, it will usually fire at the top of the peak, and it will, if we do that a number of times, we just record that over then it will sometimes fire a little bit away from the peak, but mostly it will fire at the top of the peak. So we'll build up kind of a histogram in this little hair cell here, and it turns out that the histogram is a representation of our fine structure, representation of our waveform. This is the waveform. Say it's 1000 Hertz tone or something, so it goes up 1000 times per second from here to there, and all of the spikes happening at the top half, or at the maximum of the amplitude. No spikes happening at the bottom here. Why is that useful? Because every time a spike happens, we know something about the stimulus, namely it was here. So this is called phase locking. The spikes are actually locked in phase or locked in time with the phase of the stimulus. And we can use this information because it is just a representation of the time. And this is how we get to all of the time or the temporal aspects of hearing. So here's another way of looking at it, just more schematically. We've got our sounds. Let's say this is a 1000 hertz tone, and we measure when our auditory nerve fiber is firing. It would be here and here and here. It's not firing at every peak. It's skipping that one because nerve fibers are just simple neurons. They have a refractory period. They're getting tired after a while, so they cannot keep up high firing rates for a long time. But if they fire, they will fire at a fixed point during the amplitude. And if we do that more often, we always get a multiple of the periodicity. So either here 2 periods or 1 period. If we respond or if we measure an auditory nerve fiber to a very high frequency, above 5 kilohertz, then these spikes would be completely random. They would not be locked to the frequency anymore. We'll investigate that in a second. If we have that's what's called period histogram. So this is the histogram that we just built up. So if we count the number of spikes in a response to waveform, we get a very good, you can call that a sampling. So what the auditory nerve does is it samples the waveform very accurately, but only, you see that, the top half, not the bottom half. It cannot code for negative half waves, it can only code for positive half waves. It's called half wave rectification, but that should not concern us. Now, it has been measured in many nerve fibers, psychophysically as well, but here specifically for a cat and a guinea pig, because it's difficult to do this experiment in humans, obviously. They measure at the function of the frequency is 100 hertz, 1 kilohertz, 10 kilohertz. Each of these dots is one nerve fiber, and what has been measured is how well this function is true. So how often the spike happens at exactly the highest point. If they only ever happen at the highest point, we give that a number 1. And if they are completely random across, so they fire sometimes here and sometimes here and sometimes here and sometimes there, we call that a 0. That's then what is called a synchronization index, measured between 0 and 1. And you see that at low frequencies, up to 1 kilohertz, this is perfect. Well, as good as it gets. And then it drops at around 5 kilohertz, depending on the animal. The guinea pig here at about 1, 2, 3 kilohertz, the cat about 5 kilohertz, the barn owl about 10 kilohertz. It will drop to 0. So neurons cannot follow the time structure of the stimulus anymore. It's called end of phase locking, and it is the end of all temporal information about the stimulus. So something happens in between, well, something is different between stimuli below 2 kilohertz or above 2 kilohertz. Do you know what, perceptually? It's the end of musical perception for us. And we'll find out about it more about when we talk about pitch perception. But it's the end of a lot of things. It's the end of space perception, and it's the end of musical perception for us. So all of our music happens way below that frequency. What I haven't got is data actually where the human is, but the human is comparable to the cat. So the cat is more accurate, as is the human, and we have, our threshold is about 4 to 5 kilohertz here, I should say. Okay, here's a little summary about this phase locking. So a given nerve fiber tends to fire most often at a particular point or phase of a low frequency tone. At about 4 kilohertz this ends for us humans. Do you know what the highest frequency on a piano is? 4 kilohertz. That's why. Because above that it doesn't make sense to play music anymore. We don't perceive it as musical. Corresponding to the end of musical pitch perception. Fibers fire with equal probability in every part of the stimulus. Yes. Why is that physiologically? It's probably due to the reduction of the AC receptor potential. You remember very first lecture when we showed the AC and the DC component at about 4 kilohertz. The DC component becomes dominant and the AC component becomes negligible, and so the hair cells cannot, well, the stereocilia cannot cope anymore. The outer hair cells cannot vibrate. Well, no, they still can. The inner hair cells mostly cannot keep up with the AC component, and so the nerves are not able to fire on the frequency anymore. Okay. Yes, more details on that next week, because when we talk about pitch perception that becomes relevant. But here it explains for us how the temporal processing comes into play neurally. So we talk about perception always, we talk about pure tones and gap detection and things, but what we really perceive are the spikes of neurons, and the neurons must somehow be coding the temporal properties of the sound. And of course we don't not only having one of these, we've got lots of these, and this gives me opportunity to explain what the volley principle is. Now here are 8 different fibers, and here's one stimulus, and each of these fibers fires at some point, and then it's quiet for a while because they're tired. They cannot fire more than 200 times a second. They will then adapt over time. We talked about adaptation, but even after they adapted, if you have lots of these fibers together and you just add them up, this is the yellow line here at the bottom, see what happens? The yellow line is a perfectly accurate description of the stimulus, because you get one spike every time the stimulus has a maximum. Even if you look at each individual fiber, you don't necessarily see that, because each individual fiber might take a few milliseconds, a few tenths of milliseconds before it can fire again. So that's the volley principle, which means it's population coding. We need a whole population of these auditory nerve fibers to code the whole stimulus. That's why we have 30,000 of them. Okay. So here's a little schematic figure of that. Just this is the unrolled basilar membrane, and here's our inner hair cells. For example, at this point, energy flows through the basilar membrane. Here's the maximum, and here's our hair cells. And each of these hair cells has around 10 auditory nerve fibers, and each of these auditory nerve fibers fires face locked to the stimulus. So you can see that here. That's basically a summary of everything that I said. Here's the face locking of each cycle, and in sum they do a volley principle, and you get most of these spikes will always be at the maximum of their stimulus. So this is how temporal coding happens, and if we put all of these together, then there's a nice figure I just like to show. If this is the spectrogram of a sentence. Joe took father's green shoe bench out. Joe took father's green shoe bench out. This is a spectrogram, and here are 1, 2, 3, 4, 5, 6, 7, 8 nerve fibers recorded at different frequencies, 400 Hz, 1000 Hz, up to 4 kilohertz. You see that they all respond in their own way. So these are just 8 out of 30,000. Imagine if you have 30,000 of them, you get even more information. But you see that these are tuned to their BFs, and they code, for example, this green, which is a diphthong. It goes up in frequency. It's nicely coded, not by one, but by all of them together. So this is this population coding. You need to integrate over various frequencies and over various time to make sense of these. This is a nice illustration of how individual neurons do parts of the workload, and all together they share the workload to code the whole of the stimulus. I think I show that figure again when we talk about speech. Okay, here's the effect on hearing impairment, or noise-induced hearing loss on the temporal fine structure. And I apologize for the quality of that. This is just taken from Brian Moore's book. But I want to point out the difference between these two figures here specifically, because the experiment that has been done here, they have measured in animal, and I think it was cat, if I'm not mistaken. Correct me if I'm wrong. Lots of neurons, auditory nerve fibers. So these are about 50 or something, and they measured the vector strength. So all of what I just said was basically only to introduce or to explain this one figure, because you need to know and understand what the vector strength is in order to understand what hearing impaired people suffer from. Because what the take-home message, before I explain it, take-home message is hearing impaired people suffer from a reduced vector strength. That's what we can conclude. So, what's the experiment? You take a cat, a normal cat, and measure as a function of the characteristic frequency the face locking. And I said it just breaks down here at higher frequencies, and that's all fine and good. That's what we know. And now you take cats and do a noise damage to them. And I don't know the details of that, but that's usually 6 hours at 130 decibel. They have a noise trauma. And then you do the same experiment again. So now they have a noise-induced hearing loss. All sorts of things have gone wrong. We know from last lecture, yeah, they have a reduced tip links, they have reduced stereocilia, they have all sorts of cells missing. But if you measure the same experiment in quiet, you don't see any difference. The face locking of the neurons is still exactly the same as in normal people. That has thrown researchers out for the last 20 years. How you not see a difference in the response of nerve fibers, even in noise-induced noise-induced hearing loss. Now, obviously, I have a point somewhere here, and the point is in the other slide. If you measure the same experiment in background noise, so you have a stimulus, a pure tone, and you measure the face locking, but you add noise, then both of these are reduced. That's what we expect, but the noise exposed fibers are specifically reduced. And that is the current research, what we can do about that, because that's what's called hidden hearing loss. Yes, it's hidden because we can't see it in quiet. But in noise, we do see a difference in the response of the vector strength, of the face locking. And to give you a short answer, what I think what's happening, I haven't got much evidence, but in general, is that only a specific kind of nerve fibers is affected first, namely the high threshold with the wide dynamic range. The low threshold ones, the ones that will respond in quiet, are still working, but the high threshold ones, the ones that code the temporal processing, these ones are the ones that are necessary for threshold. These ones are the ones that are necessary for temporal detection, and they are specifically damaged. So this is fairly 2012. This is fairly novel results that haven't, that are beginning to be digested in the research area to maybe understand better what's happening with hearing loss. Okay. And now to something completely different again, just 2 minutes, about combining information over time. As a very, very schematic illustration of what I actually said at the very beginning. If you have a stimulus, we have two ways of interpreting what our brain does to it. If it's long stimulus, why do I need long stimuli? Why not just a short stimulus? Well, there are two ways of interpreting that. One is, if I have a long stimulus, I can look several times at it, or I have an integrator that takes a longer time to get something out of that. Give me one keyword where this is actually important for our audiological day-to-day work.
The firing rate?
No? Threshold, sorry. It's so simple I don't... you're probably not even thinking about it. The actual threshold, PTA threshold, pure tone audiometry. What are we measuring? We're measuring a threshold perception of a tone in quiet. And I mean, leave away all of the problems that we discussed with the signal detection theory, which basically means this is a pretty meaningless way that we're measuring anyway. But if we measure it perfectly with the, say, two alternative forced choice, adaptive procedure, we get a really good result. We get the reduction of a threshold with the duration of the stimulus. Now this is an integration effect here. Long integration. What happens if I get a very short term, 10 milliseconds, let's say we've got 29 decibel for this specific kind of experiment, these kind of people, but if I double the energy of the stimulus, I would expect a halving of the threshold, or in other words 3 dB less. And that's exactly what we find. We get a 3 dB decrease in threshold for a doubling of energy, up to about 100 milliseconds. So the auditory system integrates information over time, up to 100 milliseconds, and then it's about constant, somewhere around there. Now, in PTA, what do we use?
Few times.
Yeah, how long?
Oh, about 3 seconds maximum.
3 seconds maximum, yeah.
1
So
to 3 seconds.
1 to 3, we're way, way, way in here. So we don't consider that. Why do we do that? Because we don't want to consider that. We explicitly, well, I don't know. This is not written down anywhere. I'm just making that up. But if we had the effect also of the duration to consider, it would become much more complicated. So we're trying to take a stimulus that is not complicated by the duration integration, and that's why we take a long stimulus. However, there's a research question here for you. Is that the right thing to do? And here, spoiler alert, I don't have the answer to that, but I challenge you as masters of audiology to think about that. Is that the right thing to do? How many stimuli do you know in nature that are 3 seconds long? Exactly 1 in the pure audiometry. Normal stimulus are in the range of tens of milliseconds. Speech stimuli are rarely longer than 30-40 milliseconds, because then they fluctuate again. So we're talking about the perception here. And we need to consider this as an effect of compression of the amplitude of the modulation for hearing impaired people. So by being over here, we completely ignore people's problems with compression, which means we fit hearing aids to something which potentially is completely irrelevant for them, namely long pure tones and not speech. So I've come to the conclusion that the complete process of PTA is fairly, sorry, I'm on record here, I shouldn't say that, but you can follow that yourself. Yes, so I'm quite skeptical about that. Okay, and last thing for today, just to make something which I'm really interested in. I'm really research-wise, I'm really interested in this whole concept. Not only for hearing impaired people, but for the perception of sound and what makes sounds interesting, and how do we understand speech in noise specifically. Not only hearing impaired people, normal people. Because if we understand normal people, we can maybe understand also how we can automize the process and how we can make it easier to understand for everyone.
Sorry.
Yeah.
I have a question about the content you explained previously. If you fit hearing aid based on SRT threshold, this
SRT, speech
SRT.
reception threshold.
Yeah.
Okay, let's leave that for in 2 weeks when we talk about speech. Okay, then we define what SRT is, and we can. That's an interesting aspect. Exactly. Maybe a very good alternative. That's right. That's right. That's very good. Keep that please in mind. Okay, but here we're talking about only auditory, well, the pure tone thresholds, because that's the one that we all use in practice. Okay. Modulation filter bank, and this is more like an excess. No, what's the word? An add-on. don't worry, this is not basic stuff. This is quite advanced auditory research, which is not so in the mainstream, but it's really interesting because imagine you do an experiment where you have not only one modulation, but you have two modulations. And you detect not the stimulus, but you detect the modulation. So that's what we did with the TMTFs, temporal modulation transfer function. You detect the modulation of a sound. And if you do that, you want to detect one of the modulations, it turns out that you can mask this detection of a modulation with a different modulation. Very analogous to masking a pure tone with a noise, or masking one pure tone with another pure tone. We can also mask one modulation with a different modulation. And if you do that, and you don't have then the frequency up here, but you have the modulation bandwidth, you get a result which grows 3 dB per octave, and then it stays constant. Very analogous to the experiment that we actually did, very broad experiment that we did to discover the width of the auditory filters. So it turned out that there's not only an auditory filter, there's also an auditory modulation filter. An auditory modulation filter. So the modulation of a sound is apparently equally represented in the auditory system than the actual frequency itself. And here's another result, just to make that clear. We've got here, this is the schematic one. This is what we already heard. This is the 1000 Hertz tone modulated. We have a different modulation on there that masks this modulation. And if we measure the perception of that, so we're not here, this one is not the dB perception threshold, but the dB modulation depth perception threshold. So 10 Hertz, we're very, very good at modulation detection here for this filter, for example. But this is masked by other modulations. So if we go outside of our modulation filter, we get higher masking. So you recognize these filters. They look very similar to auditory filters, but they've got nothing to do with auditory filters. They're the equivalent of that in the modulation domain. And how that happens is stuff for a different lecture. But the fact is it does happen. And what people more and more believe, and maybe I should put one paper up there, which has just been published 2014, this year, is that more and more people believe that this modulation is actually more relevant for masking, not only than we ever believed, but potentially more important than the energy. Just to make it clear, this is not something that happens on the basilar membrane. The basilar membrane doesn't even see that kind of modulation because they just filter with the auditory filters. Everything that we said is still true. But if we go higher up in the brain, our brain seems to be tuned more and more to the modulations. Low frequency modulation, here we're talking about, well, few hertz, up to 100 hertz. This is the modulation of our fundamental frequency of speech, down to the prosody of what happens to vowels and consonants. That's the kind of frequency that we're talking. And the brain is highly tuned to analyze that. And the conclusion that some researchers do now is that masking or understanding speech and noise has less to do with the noise that is masking, like the forward masking that we're talking about, but the actual modulations in the noise that mask the modulation in the speech. And this is an open research question, really. This is this year, and will certainly be coming in the next few years or something, when we have to understand that and maybe, well, solve it. Maybe it's not true, I don't know. But at the moment, the message is modulation is actually worse for understanding, for masking. I say, well, modulation masks more than other things, than energy. And if that is true, then practically all of our approaches to reduce the noise in hearing aids, say, are misinformed, to say the least, and not working very well, which they don't. And maybe that's the reason. Okay. So, here's the consequences with which I leave you, and maybe to think about for the tutorial tomorrow on Wednesday. Oh, sorry, tutorial on Wednesday. There isn't a tutorial tomorrow because we have this guest, Roy Patterson, and I invite you to go to the seminar, but we will do the tutorial on Wednesday morning instead. So what you can think about, why do we measure PTA for long sounds? That's already the question that I raised. And maybe you review that and try to understand where I'm coming from, that we might actually measure the wrong stimuli here. How do you think various aspects of masking that we could have covered might affect the speech recognition in quiet and in noise? That's what I've tried to shed some light on, because there are some differences between speech in quiet and speech in noise. How do you think speech perception performance would vary in people with different amounts of remote masking, temporal masking, forward and backward and combined? Can you think of ways to improve speech recognition in people having problems arising from remote and temporal masking? That's an interesting one. There are a few PhDs in that question. But yes, I'll leave you with that. Thank you. Or are there any questions? General consumption.